All right, once you have completed the Krebs cycle, the next part of respiration is the respiratory electron transport chain. And that takes place in the inner membrane of the mitochondrion, the cristae membranes. And the molecules that do this electron transport chain are located in that position. Okay, now the reduced NAD that you make from Krebs cycle will enter the electron transport chain. And as it travels down the electron transport chain, energy will be lost, and at 3 positions along that route, enough energy is lost to make an ATP. So for every reduced NAD that goes in the top of the electron transport chain, you get 3 ATPs. Now at the bottom of that electron transport chain, once it's been milked of all its energy, the electron drops off and binds to oxygen, or the electrons drop off and bind to oxygen. And when they do, the result is the production of a molecule of water, because once the oxygen picks up electrons, it will also pick up 2 hydrogens, and the result will be a molecule of H2O. Now I'm going to take you through the electron transport chain and show you how the thing works. The orange circle is an electron, or actually a pair of electrons. The reduced NAD will pass on to the electron transport chain, and it will be passed to molecule FP, as you will see in the next slide. And here you can see the molecule FP picking up the electron from reduced NAD. Here you can see molecule FP passing its electron off to Q, and as it does, enough energy is lost to drive the production of ATP. And so you've now made 1 ATP molecule. And Q passes the electrons off to cytochrome B, C1. Cytochrome B, C1 passes the electrons off to cytochrome C, and once again, during that passage, enough energy is lost to drive the production of another ATP. Cytochrome C passes the electrons off to cytochrome A, A3. And finally, cytochrome A, A3 passes the electron off to oxygen, which will eventually make water, as you'll see in the next slide. And once again, for the 3rd time, you have a sufficient drop in energy, a sufficient loss in energy, so that the electron drives the production of a 3rd ATP molecule that comes from NAD reduced. And here you can see the electrons ending up in a molecule of water. Now, I haven't shown you this yet, but reduced FAD also donates electrons to the electron transport system, but it doesn't do it at the same place as reduced NAD. It does it a bit lower down the chain, and consequently it skips producing the first ATP. The result is that where it goes in, which is at molecule Q, it will travel down the electron transport chain and drive the production of 2, not 3 ATPs, and the electrons will end up in the same place, binding to oxygen and forming water. Now we'll discuss the process by which the ATPs are made by the electron transport chain. This process is called chemiosmosis. And it turns out that some of the molecules in the electron transport chain pick up both electrons and hydrogens. Other molecules in the electron transport chain pick up just electrons. So, if a molecule that picks up electrons and hydrogens passes off its electrons to a molecule that binds to only electrons, the hydrogens have to go somewhere. And invariably what they do is they end up in the space between the inner and outer mitochondrial membranes. The hydrogen ions are picked up from the matrix. The result is that these hydrogens get pumped by the electron transport chain from the matrix of the mitochondrion into the space between the two mitochondrial membranes, the inner and outer. The result is a buildup in hydrogen ion concentration in between those two membranes and a reduction in hydrogen ions within the matrix of the mitochondrion. When you have this kind of situation, what you end up with is something called an electrochemical gradient, which is the strongest kind of gradient that you can have. It's a gradient in both concentration, as you can see from the drawing, and also charge. And so those hydrogen ions want to get back into the matrix as soon as they can. Unfortunately, they cannot pass through the membrane because they are small and charged and consequently can't pass through the membrane. So they have to find a pathway through. The pathway through is that the lollipop structures that we discussed earlier, which are now called ATP synthetases. The ATP synthases, not synthetases, sorry, synthases, the ATP synthases provide a passageway through which the hydrogen ions can re-enter the matrix of the mitochondrion. And along the way, they drive the production of ATP by the ATP synthase enzyme. Now, the way this works is very similar to the way a hydroelectric dam works. A hydroelectric dam is placed in the middle of a river. Water builds up behind it, so the water concentration is high behind the dam, but it is low in front of the dam. What these hydroelectric dams do is they allow the water to flow through the dam through a passageway, and as the water flows through the dam on that passageway or along that passageway, it turns a turbine. Now, as the turbine turns, one end of the turbine is wrapped in copper wire, and that copper wire is turning in the middle of a magnetic field because there are magnets surrounding the stem of the turbine. And as the copper wire turns in that magnetic field, it generates an electric current. Sort of the same thing is happening in the ATP synthases. As the hydrogen ions pass through, the head of the ATP synthase, the lollipop on the stick, turns, and it turns in sort of ratcheted turns of 3, and every time it turns, it spins off an ATP molecule. The result is the production of ATP using the electrochemical gradient of hydrogen ions to drive it. And that electrochemical gradient was built up by the electron transport system that pumped the hydrogen ions from the matrix to the space between the two mitochondrial membranes. This is another picture of the electron transport chain, and it shows the flow of the electrons through the electron transport chain, and it also shows you the ATP synthase molecule on the right. Now, the production of ATP by the electron transport chain and driven by the buildup of the hydrogen ion concentration gradient, the electrochemical gradient, by chemiosmosis, the process of making the ATP in this way is called oxidative phosphorylation. This is a negative staining electron micrograph of lollipop structures, or ATP synthases, on the inner mitochondrial membrane, the cristae membranes of a mitochondrion. You can see that they do in fact look like lollipops, and we're going to show you a little more of their finer structure later on, but this is what they look like, and that's where the name lollipop structures came from. This is a higher magnification picture of a single ATP synthase molecule, a single lollipop structure. This is a schematic drawing of the ATP synthase molecule, and what will happen is as the hydrogen ions travel through it, it will turn in the direction you see with the arrow. So it will revolve on its stalk. It'll turn around on its stalk in that direction. The dotted line shows you the passageway through which the hydrogen ions will pass. This is a picture of the chloroplast ATP synthase using an atomic force microscope image. And you can see the roundness of it and you can see what it looks like there. And we'll have more to say about that later. This is a higher resolution view of the ATP synthase in chloroplasts showing its 14 subunits. Again, this is an atomic force microscope image. This is a picture of vesicles made from the inner mitochondrial membrane, and you can see the ATP synthase molecules on there, the lollipop structures, and this is again an electron microscope image, as you might imagine. This slide shows you the 3 steps that the lollipop part of the ATP synthase molecule go through to produce the ATP. There's a 3-step reaction that goes on, and as that reaction occurs, the head of the ATP synthase molecule actually turns around in 3 steps, and after the 3rd step, it will release an ATP. This is an experiment that shows that the head of the ATP synthase actually turns. What they did is they took the lollipop head of the ATP synthase and they attached it to a glass cover slip. They then were able to attach an actin filament to the top of the head, and then they gave it a proton gradient to drive it, and they could see the actin filament turning around. Now you might say, well, wait a minute, the head wasn't turning. No, what happened was the head was attached to the cover slip, and the part, basically the part of the yellow part there that you see in the slide, was turning while the head itself was immobilized. Normally, however, it would be the head that turns while the yellow part remains stationary, and they could actually see in the electron microscope the actin filament going round and round and round. Here you see how the turning of the ATP synthase head will pick up the hydrogen ions in the space between the 2 mitochondrial membranes, and as it turns, will eventually dump the hydrogen ions back into the mitochondrial matrix once it's made its way all the way around, once the turn has gone all the way around.